Okay, let's get started. Hello, everyone. It's my great pleasure to introduce Professor Chris Zahasky from University of Wisconsin-Madison. Chris actually did his undergrad in our own department, so he's our alumni. So I think special meaning for you to come back. Chris did his PhD at Stanford, and after that stayed, I think, a little bit as a postdoc, and then did also another postdoc at Imperial College London, and then started his tenure track a few years ago at UW-Madison. Today, Chris will tell us about solute and bacteria transport to geologic porous media using cutting-edge methods. So, floor is yours. Thank you.
Cool. Yeah, thanks for that introduction, Peter, and thanks for the invitation to be here. As Peter mentioned, I finished my undergrad degree in this department and actually did my senior thesis at SAFL. So it's great to be back, and I was sort of going back through the archives and found a picture from some corner of SAFL where I actually did these experiments where we were trying to understand the formation and propagation of fractures in anisotropic materials, specifically these clay cakes. And I don't want to get into the details of this, but in sort of reflecting on this, there were some sort of research themes that evolved or sort of that came out of this that I think you'll see sort of throughout the work that I'm going to present here today, and that have really influenced the type of research that we're doing in my group at UW-Madison. And so these three sort of themes are, first, that experiments are really hard, but they're really essential for being able to isolate processes or mechanisms so that we can quantitatively understand these systems better than we might be able to through purely observations in nature alone, by being able to remove some of the other variables and signals that can influence our ability to quantify these processes. So that was sort of theme number one. Theme number two that I'll talk about quite a bit more today is the power that images can provide to understanding really complex systems, being able to not just get a single measurement, but to be able to actually parameterize two-dimensional or three-dimensional properties that are important to a system, and essentially translating these images through math to some of these important properties. So in this case, we were trying to calculate strain, but what I'll talk about today is using three-dimensional images to characterize permeability and then also bacteria attachment behavior. And then the third research theme that sort of came about, not just through this research, but through associated fieldwork and my general education in geoscience, was the ubiquity and sort of intrinsic nature of heterogeneity in the subsurface. We have sedimentary features and structural features, and all of these things make it so that we have variation in fluid flow behavior from the pore scale up to the basin scale. And so if we really want to understand and be able to forecast different flow and transport problems, we need to be able to characterize and understand the role that this heterogeneity has. And this is, I think, one of the best illustrations of this. This is a photo that I took a few years ago at an outcrop in Green River, Utah. And what's showing up here, the black areas, are areas that were stained by oil as oil percolated through this sandstone reservoir millions of years ago. And then this reservoir was exhumed, and we can go out, we can essentially trace out the pathway that oil took as it was migrating through this formation. And it's really striking because you see, the complexity of this flow behavior that occurred in the subsurface and the role that different sedimentary features had and structural features like deformation bands or fractures had on the flow and transport in the subsurface. And I think this essentially really highlights, the impact and the importance of being able to characterize the spatial variation in these different fluid flow properties if we want to understand flow and transport processes in the subsurface. And so you can see that even at the millimeter or centimeter scale, we have this heterogeneity that's actually controlling a lot of this displacement behavior all the way up to the meter scale. And so the first thing that I'm going to talk about today is just that, essentially being able to take image-based information and characterize permeability in 3 dimensions, because whenever we come across some flow or transport problem, we almost one of the first things we want to do when we're building a model or trying to describe how fluids flow in the subsurface is we want to have some information about permeability, the resistance to flow as fluids are flowing through a porous or fractured media. And typically, if we want to get really good data, we go out into the field, we drill some boreholes, we get some core, we bring it back to lab, and we'll go in and try to measure, we'll measure a bulk average permeability, essentially flow fluid through at a certain flow rate. We'll measure the pressure drop and use Darcy's law to back calculate permeability of that sample. But that really only gives us the permeability at one particular location. And what we often want when we're trying to account for things like this variability and uncertainty in building these models is we want a distribution of these values in order to understand some of the spatial relationships of permeability, but also understand some of the statistical distributions that we have. And so what I'm going to talk about today is how we've gone about doing that. And so just to provide a little bit more background, you all have fantastic imaging, X-ray CT imaging facilities in this in the Department of Earth Science here. And this is awesome for being able to essentially highlight and understand the structural variation in different types of rocks. So if we take this sandstone core sample that I'm showing here and we look at this this X-ray CT image on the right, what we see is variation in essentially electron density or sort of analogous to material density. And so what this allows us to do is make out some of these structural features like axial parallel bedding or some of the subtle cross bedding features that we have at the top of the sample. And from that, we can go and understand a little bit about the structure and what the permeability structure might look like. We can also use X-ray CT to go and map out in 3 dimensions the porosity variability by taking X-ray CT images under dry and under water saturated conditions. And for these reasons, that the CT is really giving us some information about density, it's really ideally suited for mapping the structure, mapping porosity, mapping fluid saturation variation when we have strong densities differences in our fluids. But it's not so good at being able to estimate permeability variation. Because if we want to go from something like porosity to permeability, there are literally dozens, maybe hundreds of empirical relationships that go and can relate porosity to permeability, and they're always dependent on lithology, cementation, and the specific geology of the core samples. And so if we want something that can distinguish and help us map 3-dimensional permeability in carbonates and these relatively homogeneous Boreas sandstones, and then in much more complex sandstones, we need to, these are all going to have different empirical relationships between porosity and permeability. So we need, another method to be able to get at permeability of these samples. And so a few years ago, what I started working on was using positron emission tomography as a complementary imaging technique to X-ray CT imaging. And with this imaging technique, essentially we rely on using radioactive tracers or radioactive isotopes attached to species of interest that we want to quantify in geologic materials. And so the way that this works is we have some positron emitting radio tracer. As positrons are emitted, they annihilate and they produce these coincident photons, which then can be detected with this ring of photon detectors in the PET scanner. And over the course of hundreds of thousands and millions of these decay and detection events, we can map in 3 dimensions the distribution of this radioisotope or this radio tracer as it is being transported through a geologic material. And these photons are very high energy, so it's really ideally suited for dense geologic materials that have very high density. And so what we've done at UW-Madison is build a mobile experimental platform that allows us to be able to go to different imaging facilities to be able to do these types of experiments. And this is just a picture of a few happy graduate students plugging away in the lab, collecting some of the data that I'll show today. If we go back to this same core sample that I've been showing a few slides ago, what this data actually looks like is shown on the right. So typically, a common type of experiment that we'll do is we'll do a pulse tracer injection. Essentially, we'll load this core into a core holder so that we can replicate subsurface pressure and temperature conditions. The cylindrical faces of this sample are sealed, and we inject fluid from one side and it flows out the other side. And so we'll inject, for example, a radio tracer where the intensity of the red is showing this conservative radio tracer distribution, the concentration or the essentially radio intensity per volume. And then we'll go and displace this tracer with water. And then what we can do is we can image again this distribution of how this tracer is flowing through this three-dimensional geologic sample as a function of time. And so we get really pretty, three-dimensional distributions of this tracer. But what we also get here is within every single one of these voxels or these three-dimensional pixels is we actually get breakthrough curves. We can measure the concentration as a function of time, which is what I'm showing in the plot on the left, for every single voxel in this material and or in this image. And so what I'm showing here are three different voxels at different locations and the corresponding breakthrough curves with the solid gray line illustrating the snapshot in time that corresponds to the image on the right. And so as we have this tracer that's being transported through our system, we go and we have, or our core sample, we have these corresponding breakthrough curves that correspond to different voxels. And so this provides an absolute immense amount of information to be able to quantify a whole range of transport properties like dispersion and advection and some of the variation in those behaviors across the sample. And as I'll show a little bit later on in this presentation, we can also go and attach these types of radio labels to other species of interest like bacteria, so we can also get information about bacteria distributions and attachment. And so we've done these types of experiments in all sorts of cores, sandstone cores, carbonate cores. And the first thing that I want to present today is addressing this first question, which is how can we take these rich four-dimensional data sets and turn them into parameters that are important for flow and transport models. Essentially, how can we take this information and map the three-dimensional permeability that creates this this variation in this advection behavior that we observe at a local level within these different core samples. And we've developed a couple different methods to do this over the last couple of years. The first is using convolutional neural networks, essentially an image-to-image regression model that takes PET imaging data in and produces a three-dimensional permeability map out by essentially training this model in reverse, as I'll talk about in a few slides, and then using and comparing this to a more classic numerical inversion technique called Ensemble Kalman filters. So this is more of an iterative approach where we go and take our observational data, our PET data, and we compare that with a bunch of different realizations of that permeability field, and we update these permeability maps and run lots of forward transport models to on these updated maps. And so I'll briefly talk about the Kalman filters a little bit later on, but what I want to focus the first part of this talk on is this application of convolutional neural networks to be able to take this three-dimensional or this four-dimensional data and map out the permeability in 3 dimensions. So in order to do this, I sort of broke this up into steps. There's kind of 4 main steps in the application of these convolutional neural networks to be able to perform a task like this. The first is that we need to ideally process the data in order to reduce the dimensionality of these data sets. These are massive 4D data sets, and it would be ideal if we could at least get this data down to the same size as that 3-dimensional permeability map that we're trying to get as an output. Then in this case, we're doing a supervised learning approach. So we have to generate a whole bunch of data in order to be able to construct and train these networks. And then we validate our neural network, not just with test data or synthetic data, but actually with this real experimental data. Okay, so step 1, reduce the dimensionality of the data. Well, if we go back to our imaging data, again, we have breakthrough curves at every single voxel. And one of the nice things that is that there's this rich history of different types of analysis that people perform on breakthrough curves in order to understand something about the transport behavior. And one of those analyses is quantile analysis, which you can think of as essentially a measurement of how much tracer has passed by a location at a specific time. So for example, the 0.5 quantile is the point in time at which, in this particular voxel, 50% of the mass or radioactivity in this case of that tracer has passed by a particular location or that voxel in this case. And so what this allows us to do is we can essentially take all of this rich time series data, and we can collapse that down into essentially an arrival time map in these particular core samples. And in addition to converting this from a 4D matrix of information to a 3D matrix of information, there's a number of additional benefits that were outlined in sort of the early development of this this analytical sort of classic hydrologic approach. And one of the benefits is that we're averaging out some of the voxel level imaging noise. So anytime we're making measurements in the lab, there's always some uncertainty and noise that's interfering with our signal. And essentially by integrating over these breakthrough curves, what we're doing is we're essentially averaging over the subtle noise at each time step, and it allows us to more accurately quantify a specific time of arrival. The other thing that is really useful is that it allows us to neglect dispersion. And this is really important for two reasons. One is that, for example, those carbonates can have very long tailing behavior that can make it so that we can have very sample specific dispersion behavior that would make it difficult to build an algorithm that can robustly handle a wide range of different types of rocks. But the other advantage is that when we're doing generating numerical training data, it allows us to not have to worry so much about numerical dispersion, which is a numerical challenge when we're often that arises when we're trying to model transport efficiently. And so being able to neglect that is valuable. And then the other really important thing is that enhances the generalizability of this algorithm is that it allows us to have some independence of inlet concentrations or pulse volumes or some of these sort of experiment specific examples. And so when we go and calculate, for example, this quantile arrival time map, this is what we get. This makes sense. We have earlier arrival times indicated by the yellower colors, and later arrival times towards the outlet of the sample indicated by the higher arrival times or the darker green colors. And we do some dimension. We can normalize the time component of this by converting the time in units of seconds to pore volumes. So this is a way to non-dimensionalize our time so that we essentially go from 0 at the inlet to 1 near the outlet in a homogeneous sample. But what you'll notice is we have this linear trend due to this overall sort of bulk one-dimensional nature of these experiments of flowing tracer from inlet to outlet. And so what we thought was, well, maybe we could go and subtract this linear signal that arises, that sort of underlies the sort of overall trends within this arrival time map. And so that's what we did. We call this our arrival time difference map. And the benefit of this, by subtracting that linear trend, what we're doing is we're amplifying those subtle variations and that essentially the effective the transport rates in different parts of these core samples. And the other benefit that this has is that it also has more structural similarity potentially to the underlying permeability fields in these samples. Not always, but in some cases, our permeability doesn't have this linear trend to it. It has a trend that looks more like this. And so it just aids in that image to image regression training process to have some amount of structural similarity between our input data and our output data. And so we look, if we want to be able to interpret these pretty colors, essentially what you can see is that the greener areas, in this case, correspond to higher advection rates. So they're faster than average pore volume arrival point. And the pinker areas are areas where we have slower advection. And so we might expect these areas to be a little bit lower permeability. And those are in these arrival time difference maps illustrated by negative values. So these are areas where advection is occurring slower than average in a homogeneous sample. And greens are areas where we have faster than average advection in relative to a homogeneous sample of these dimensions. So what we have now is we have this arrival time difference map where we compressed this 4-dimensional information into a 3D matrix that is the same size as the permeability map that we're going to try to extract from these samples. So the second step in being able to apply these convolutional neural networks is we need to be able to train these networks, and we need to do, in order to do so, we need to have a whole bunch of data. And so what we did is we generated 20,000 synthetic 3-dimensional permeability fields. There's some awesome open source tools to be able to do this really efficiently. And we sampled a massive parameter space when going and generating these permeability fields, because if we want to be able to invert the permeability of a core sample, that the characteristics of that permeability of that core sample must fall within this parameter space that we're training our neural network on. And so we have a wide range of different correlation lengths, correlation orientations, mean permeability values, so that we can handle, again, a wide range of different core samples. And what these maps are just showing are log permeability distributions of just 3 random permeability fields from this set. So once we have these permeability maps, then what we want to do is we want to get something that's similar to what we've been able to extract from our PET data. We want to be able to calculate essentially a normalized arrival time map. And so to do that, we use a pretty traditional numerical modeling approach. USGS has this MODFLOW numerical model and MT3D for modeling the transport. And they have these great sort of Python wrappers for this modeling framework, so that we can really efficiently, run 20,000 forward models on all of these synthetic permeability fields, so that then we can calculate a corresponding arrival time map. So this is what we're in essence going to do, is we're going to train our network to take this as input and be able to essentially run this process in reverse. And so the third step is being able to construct this network and do this specifically the hyperparameter tuning that's necessary when building these networks. So to do this, what we did is we took an adaptation of a dense net. We started with a dense net that relies on these very densely connected blocks where we have many convolutional layers, and the interconnection of these passes this information through these different layers. And I won't get into a ton of the details here, but the sort of fundamental way that these networks work is by having these convolutional layers, where these convolutions, what we're doing is we're training the weights within these kernels. And depending on the weights of these kernels are going to be extracting different types of information. So for example, this specific kernel here, if we went and took this kernel and we multiplied it across a given 2-dimensional image, like this synthetic permeability field that I generated here, what this kernel is it's a resolution enhancing kernel. The specific weights on this are it's a pretty classic kernel in sort of image processing that enhances the small scale features within this image. If we look at a couple other examples, if we have kernels with weights that look like this, where we have 1s here and 0s here, what this is doing is it's essentially enhancing or sort of extracting the vertical features of this permeability field. And so we get something that looks like this, and then a kernel that has a geometry that looks like this and weights. What it does is it extracts the sort of horizontal features at that scale within that synthetic permeability field. And so by training the weights of these different kernel layers within this convolutional neural network, what we're able to do is essentially extract these different features at different scales within this image in order to be able to learn that relationship between our input data, which is the arrival time difference map, and relate that to the corresponding permeability field that generated that corresponds to that. So for people who do this, I include some lost training curves, but essentially we break our training data set into 3 different sets. We have our training set, which is what we're using to actually update those weights. We have our validation set, which is used in the training process for calculating loss, in addition to calculating loss on the training set. And then we set aside some amount of this data that the network has never seen in order to test the network and essentially see how robust it is at predicting the permeability on a data set that it has never seen before. So the final step in doing this is being able to really validate this first on this synthetic data. So in the synthetic data, we have the benefit of knowing that initial permeability. And through the numerical models, we calculated this arrival time difference map. We put that arrival time difference map into the network, and it gives us out a predicted permeability. And then because we know what the initial permeability was, it's pretty straightforward to calculate the difference between these images. And so that's what I'm showing schematically here. We have our synthetic permeability field. We've calculated using those forward numerical models. We've calculated this normalized arrival time map. We add some noise to our synthetic data to, again, replicate the noise that is truly there when we are measuring and gathering experimental data. And then we go and we get some permeability prediction that we then can go back and compare to that initial permeability map. Similarly, this is just showing another example. So this is one of the better realizations within our test set and one of the worst realizations in our test set. But these log permeability scales are identical in both cases. And so you can see that even the ones that have a fairly low R2 score actually do a pretty good job still of capturing that structural behavior of that permeability field. Another way to sort of assess the accuracy of these is through cross plots. On these plots, these are just 15 randomly chosen realizations within that test set. And the x-axis is showing the synthetic permeability, essentially our known permeability. And then the y-axis is showing at every single voxel or grid cell the corresponding predicted permeability. And a perfect prediction of the neural network would fall on this 1-to-1 line. But overall, we can see that within our, these 15 randomly chosen test sets or test maps, that we have, really good prediction ability of the permeability. It turns out that that's actually the easy part. The harder part is being able to apply this to real experimental data. Real experimental data has imperfections in the boundary conditions and other things that we actually had to go back and train this network and develop training sets several times that accurately captured some of the experimental artifacts and experimental imperfections that occur when we're running any type of experiment. And so this step, comparing against real data, was really valuable in building up the robustness of this workflow. So in this case, again, we have our PET data. We get our experimental arrival time map, as I described earlier. We plug that into our neural network, and about 5 seconds later, we get a predicted permeability map. But how do we know how accurate this permeability map is? Well, what we can then do is plug that into our forward flow and transport model and essentially model what, based off of this permeability field, what would this arrival time map look like? And then we can calculate the differences and compare the modeled arrival time with our experimental arrival time to again be able to assess the accuracy of this prediction when we don't know what that permeability is beforehand. And so we did this again with this Berea sandstone. We did this with a range of carbonates. So this is just an example of the Edwards and Indiana and Keton. Again, we're able to compare our experimental arrival time with our model arrival time based off of those predicted permeability fields. And what we found was that this method works really well. Again, assuming that we've developed a training data set and we've trained our model on permeabilities that are representative of the samples that we're dealing with. So for example, we didn't train our network on fractured samples. And so it wouldn't be able to predict the permeability of a fracture because that network never saw that type of information. So we published that last year. And what we started to wonder is, well, okay, this method is great. We think it worked really well, but there's decades of research on these types of inversion problems, mostly at the field scale. But what we wanted to do is see, like, well, how does this method compare with some of these more traditional numerical inversion approaches? And so the inversion approach that we used was the Ensemble Kalman filter. There's a lot of papers that describe the implementation of this and all sorts of different applications. And in this case, what we did was we generate, again, 400, you can think of it as 400 initial guesses of what that permeability field might be. Using those 400 guesses, we run those through our forward flow and transport model. We compare that with our PET data. We see what the difference is between our guess and our known data, and then we go and update that, and we run that iteratively over and over and over. The downside of this is that this process is slow. It takes about between 6 and 20 hours, depending on the complexity of your sample, to do a process that once that network is trained, the network can do this in 5 seconds on the same computer. So that's one of the powerful things about again about some of these deep learning methods. But the other thing that we realized in comparing, for example, this Ensemble Kalman filter permeability field with that from our CNN was that essentially, and this intuitively makes sense, our initial guess really has an extremely strong impact on the speed of this iteration algorithm and the accuracy of that final predicted output. And so what we thought was, well, maybe there's a way we can actually combine these two approaches. We have a really, really good guess of what our permeability is from this convolutional neural network. So maybe if we just go and multiply, essentially we perturb this permeability by multiplying it by some matrix with a mean of 1 and slight value, some distribution of values around 1, we'll sort of distort this permeability field. And what that'll do will allow us to really rapidly go and generate this initial ensemble of permeabilities that we can then go and feed into this ENKF algorithm in order to potentially improve the accuracy. And so this is just showing a comparison of this hybrid method. And what we didn't really anticipate was just how much faster this would make this numerical method. Essentially, we go from having to iterate maybe 12 times before we reach some threshold to 2 or 3 times using this hybrid approach. However, the actual accuracy, it sometimes will improve with these iterations, but overall the CNN, you see it's much more similar to the CNN prediction. And so what we found was that unless you have a lot of time, essentially, the CNN actually provides a pretty accurate prediction in the first place. So this just gives a little bit of an overview of how we now are able to take geologic core samples, that maybe we get from a borehole or some other, outcrop or something. And what we can do using these methods is we can actually now map out the distribution of these permeability fields and understand a little bit about, what are the correlation lengths and things like that of these permeability structures, essentially from the millimeter scale up to the decimeter scale of our samples. And this is really valuable because if we want to go and study, different transport problems or flow problems, whether it be in energy systems or contaminant hydrogeology or flow and transport in complex natural systems, we oftentimes, once we have this 3-dimensional permeability, then we can probe a little bit deeper into these natural systems and understand more sophisticated flow and transport processes. So this sort of just illustrates a range of different other types of problems that we're working on in our group. And what I wanted to do was just transition just briefly and talk about how we can use some of these same types of imaging approaches to not just map the permeability variation that's again driven by this sort of intrinsic heterogeneity of these rocks, but now how we can go and use this type of imaging to characterize the variation, transport, and attachment of bacteria going through these types of aquifer materials. And then specifically in Wisconsin, we have a lot of land spreading of manure in a lot of areas that have fairly vulnerable aquifer, superficial aquifer systems, whether it be fairly coarse, glacial outwash materials, sandy materials in the central sands plains region of Wisconsin, or whether it be some of the fractured karst systems throughout eastern and southwest Wisconsin. And so this idea or this issue of bacteria contamination is pretty widespread in Wisconsin. And essentially what our goal is with these types of approaches is to build more accurate models that can describe that bacteria transport into these shallow aquifer systems. So just a little bit of description and background on this problem. This is just our advection, our classic advection dispersion equation. This is what we use to describe solute transport in the subsurface. And if we want to be able to describe colloidal particles, one of the sort of starting points, I would say, is colloid filtration theory, where we go and assume that we can have some first order attachment reaction term that describes the interaction and the attachment of that bacteria in our porous media. And so if we can quantify this attachment term, then that can tell us essentially how much bacteria is getting stuck and how much is going to potentially make it down to our groundwater systems. What people have realized is that obviously this is a bit of an oversimplification. And so methods and sort of ideas and models have been developed that sort of understand that we don't have a single reaction rate coefficient or a single attachment rate coefficient, but what we have in reality, just like we have a distribution of permeability and a resulting distribution of advection velocities, for example, is we have a distribution of attachment rate coefficients. So this is from a classic paper in the early 2000s showing how we can have a wide range of different deposition profiles across, for example, a half a meter. This is just showing essentially the distribution of attached bacteria from our, for example, our Earth's surface down a half a meter or so. And depending on the distribution, it can have a dramatic difference on the amount of bacteria that's getting stuck in that upper layer versus what's potentially flowing through, through the subsurface. This is just showing the suspended concentration of bacteria, again, along a column as a result of these different distributions. So we've known for a couple of decades now that these distributions are really important in being able to characterize, and describe quantitatively how bacteria gets trapped in this vadose zone and how much of it is reaching groundwater systems. But we haven't had a way to be able to actually measure this distribution of attachment coefficients. And so what we started to think a few years ago was, well, is there a way that we can use the same imaging technique that I talked about earlier, but if we could radio label our bacteria so that the bacteria is emitting, these positrons, and that we can then go and image those positrons, we'd be able to image the distribution of this bacteria as it's flowing through, for example, sand-packed columns or other types of geologic materials. And so that's what we went about doing. Essentially, this FDG, this is the most common radiopharmaceutical that's produced. It's a glucose analog. And so what that means is that bacteria will use this as a carbon source under the right conditions. And by uptaking this glucose, what that allows us to do is be able to image these bacteria as they're, again, being transported through these sand columns. So these are just sand columns. I'm showing an X-ray CT image here with a few different grain size distributions that are packed within the sample. So really subtle heterogeneity in this particular case. And then this is just a zoomed in micro CT image, again, showing that we have not just at the pore scale, we have a range of mineralogical variation as well that can be important for the attachment behavior. So now we go back to the lab and we pack a column with this type of sand from central Wisconsin, and we want to quantify that. We want to be able to quantify bacteria transport. We first do a tracer experiment. So we have a little bit of heterogeneity, so we get a little bit of a distortion of that pulse as it's again advecting through our sand column. So again, we're injecting this pulse here, it's flowing through, and I'm just showing that distribution of that tracer at different times. We can do now the same thing with our radiolabeled E. coli, except now, as you might expect, this looks very different because these E. coli, what they're doing is they're getting pushed through this pore space, but now they're physically interacting with this matrix material, this sand grains, and some of them get trapped and some of them stick to the surface. And that attachment is what we want to be able to actually quantify. And you can actually see, the sort of trail of pink that's left behind. This is, some small amount of bacteria that's essentially being stuck in these sand packs and is not flowing out of the sample like a conservative tracer does. And so, so that just sort of illustrates, again, that we know that we're attaching these isotopes to the bacteria and that we're able to image that attachment. And so what we did, I don't have time to get into the details, but by rearranging some equations, what we can do is we can essentially analytically calculate those attachment rate coefficients. So that KF term from colloid filtration theory, now what we can do is based off of the distribution of the attached bacteria after the mobile pulse of bacteria has gone through the system, is we can go and calculate the attachment coefficient in every single one of these boxes. And then what we can do is build these distributions of these attachment coefficients that then can be used to populate models to not just at the column scale, but potentially at larger scales to go and describe the variation in the way that this bacteria is essentially getting immobilized in our geologic core samples. This is a little bit of a subtle example, but this is just showing in this case, we have a layered sand pack. And so you can see it in this case, we have enhanced attachment at these layers. We're also doing experiments under mineralogical heterogeneity now, and those are kind of fun because bacteria preferentially will attach to oxides due to essentially surface forces. And so we get, much different patterns in that case too. And so what we're doing is trying to understand how these distributions change as a function of aqueous conditions. So in summary, what I've described today is how we can use positron emission tomography to provide a complementary three-dimensional imaging method to be able to characterize core scale transport processes, whether it be, again, conservative solute transport or whether it be things like bacteria. And with this, we now have these massive data sets, these massive image-based data sets, and we can use a range of deep learning approaches, numerical approaches, and analytical approaches to be able to interpret these images and understand a wide range of these fundamental processes, whether, again, it be understanding permeability variation and geologic core samples, transport and attachment of bacteria in these types of rocks and sand packs. And we're also working on a range of other important transport problems, such as channelization and fractured systems, as well as the transport of radial metals. So there are positron emitting isotopes of arsenic, copper, and zirconium that are readily available that we can actually start to use in sort of reactive transport experiments. So with that, I'd like to just acknowledge the students who did all the work. Zitong Wang was a student that I worked closely with on all of the convolutional neural network and Kalman filter development. And Vila is a graduate student group now who's doing all this awesome bacteria work. And then a range of other group members who are doing great work on all these different projects that I talked about earlier. So with that, I would, I'm always happy to hear from people if you ever have, interesting projects or collaborations, or just want to talk about flow and transport in permeable media, don't hesitate to reach out, check out our website, and or email me. All the codes for the CNN are on GitHub, and then here are some references. So with that, I'm happy to take any questions.
Great, thank you for a great talk. Time for questions.
Yeah, so hi, okay, great talk, thank you. The question I wanted to ask was relating to some of the early parts where you were using FlowPy for the like process-based forward simulations.
Yeah.
And one of the issues you kind of stated you were having was it took a substantial amount of iterations to actually kind of narrow in on a reasonable prediction, essentially. Do you think that since you're able to use these like CT methods to identify like discrete fracture networks, do you think that as opposed to using kind of like an equivalent porous media model, if you were able to go in and run those forward simulations through something that actually had discrete fractures outlined, do you think that would have cut down on the number of iterations necessary to match some of the experimental data?
Yeah, that's a good question. Well, if we're just talking about the Kalman filter, in that case, again, the number of iterations that we need in order to sort of hone in on an acceptable solution tends to increase as the heterogeneity of the system increases. And fractures are sort of the most extreme end member of, heterogeneous permeability. And so I would guess that would be a bit of a challenge. But what we have done is we've started to dabble with just how, if we could essentially feed in sort of fractured permeability fields into the CNN in order to make it so that the convolutional neural network could potentially handle that. And the challenge is really the scale at which we're sort of doing these types of models. These grid cells are about 2 millimeters each, and so you lose some of that sort of discrete fracture information. And so I think there's ways you could maybe use some of these workflows, but direct application is definitely challenging.
Hi, I have a question about the radio tracers.
Yeah.
What's like the what are the specific radio tracers and what are the half-lives?
Great question. I did not get into the details of that. So the specific positron emitting radioisotope we're using is F18, and that has a half-life of about 120 minutes or just under 2 hours. And so we like to use that tracer, we use that especially as a radio tracer, because it means that just for safety reasons, essentially everything cools down and is back down to background, radioactivity levels after about 24 hours. So you can literally, pour these fluids down the drain, a day later. Those arsenic isotopes that I mentioned, some of those have very long half-life. Very long half-lives on the order of like a day, 27 hours. And so that creates just a little bit more logistical and sort of, I'd say, permitting challenges for doing these types of experiments.
Any other question?
Thank you, Chris. It is great talk. I have a question about your estimates on the attachment rate, the KF.
Yeah.
So my intuition tells me that it would be around Gaussian shape, but it looks like power law. Why is it?
That's a good question. So in these images that I showed, this distribution, we found that log normal is what fits it pretty well. And when you look in literature, common ones that are used are log normal and gamma distributions, which you could maybe fit a gamma distribution to this. But in this particular case, what we were doing is we were trying to calculate the distribution in those 3 different layers that we had in our sample. And essentially what we found is that at the transition point between these layers, we have sort of enhanced areas of attachment as it's transitioning from like a medium sand to a slightly finer sand, and then back again. And so as a result, you get some of these voxels, sort of a smaller number of voxels that have sort of, I wouldn't say necessarily anomalously high, but sort of higher than average. And so it sort of creates this distribution that isn't fit very well with a Gaussian model.
One quick follow-up question is, how do you validate this result? Is it necessary in your opinion?
Yeah, definitely. So what we did essentially is now we have all these first order attachment coefficients, and then we actually have the spatial distribution. So we have like the attachment coefficient at this voxel and this voxel and this voxel. And what we did is we just did a fairly straightforward transport model where we have a first order attachment coefficient that's described by that specific voxel. And then we looked at the modeled attachment distribution compared with the measured attachment. Yeah, it agrees really, really well. We have a paper in review now, and I can send out preprints. I didn't have time to know all the details. But yeah.
Maybe we had one more question here.
Very interesting talk. So I have a question about the second part of your presentation about the methods, convolutional neural network methods to calculate the permeability. And from your measurement, you have methods to quantify the signal that to magnify the signal that quantitatively gives us the permeability. But for the starting part of the course, still seems like the differences still seem small. So is the model doing well for the starting part of the measurement?
You mean sort of, are there inlet artifacts within these samples? Is that kind of the question? Yeah, that's a good question. That was actually one of the most challenging things to capture, is like in a numerical model, you set a boundary condition, and that's a boundary condition, it doesn't vary, and it's not influenced by other things. When we run experiments, essentially the tracers obviously going to flow more easily into the high permeability zones. And so we had to essentially go and take our nice numerical data and sort of mess it up a bit to sort of induce that sort of artifact. But what you see is that after doing that, we are able to capture some of that variation in permeability, particularly this, this is the neural network. In some of the updates that we've done since publication to this neural network, we've actually been able to enhance some of the smaller scale features and transitions, and also improve some of those boundary condition things. So, we've restructured our neural network a bit, and it's kind of a hyperparameter sort of tuning and network architecture can have a major impact on overall quality. So,
okay, we have time for maybe one more question, if there's any.
Yeah, I want to ask about what the scenario where the hybrid approach combining CNN and the Ensemble Kalman filter performs very well compared to the other marginal approach. So one very well-known advantage of Ensemble Kalman filter is that it is more adaptable to the machine learning-based approach, because in every iteration based on the mismatch between the prediction and the data, the Ensemble Kalman filter kind of search the best candidate for the parameters like permeability. So my guess is that if the real permeability field that we want to estimate is a little away from the range of the training data set, like for the numeric neural network, then the hybrid model can kind of overcome that defect, because Ensemble Kalman filter can provide some adaptability to find the candidate that is out of the initial training data set. So how do you think about that opinion?
Yeah, essentially what we found, I mean, part of the reason there's a lot of implementations of the Kalman filters, there's sort of underlying assumptions about Gaussian distributions of the properties and stuff, or for the measurements that, they do a range of different sort of additional steps in these filters in order to sort of accommodate. And we didn't do that. We sort of put in the sort of most straightforward approach. And so what that means is that essentially when we have very strong heterogeneity in certain, or very sort of anomalously high or low permeability values, like especially in some of the carbonate cores, that the CNN does a bit better, because what that Ensemble filter wants to do is sort of revert it back to a certain distribution, at least in the way that we implemented it. So that's definitely one of the limitations there.
Okay, we have actually passed at 30 minutes, but we should end now. And thank you, Chris, again for the nice talk. And thanks for coming.